Tell us about the work you're doing on a potential vaccine. We spoke with you at the end of January. You'd already been working for a couple weeks. How is the work going so far?
Well, Mel, thank you for bringing me in. The timelines on this are we're making significant progress and very, very fast. As I told you, by the end of March, we'll have the candidate vaccine to upscale and to start working on the preclinical work to get it into the clinic. At the moment, we are working on challenge models in animals to see whether we can prove in a proof of concept, figure it out whether a vaccine could work in the challenge model in animals. And then the first step is the selection of a vaccine by the end of March, and then starting clinical trials after the challenge model, which probably will work out by the summer. We will be able to start clinical trials somewhere in early November. In parallel, we'll start upscaling as soon as we have made the selection of the vaccine in order to have large quantities available early next year. So that is the current timelines, as I indicated earlier. And I think we stay with that and we make very good progress at this moment.
Wow. And so that's even faster than I think you had even told us a couple months ago, potentially starting human clinical trials in early November. You know, originally you'd been saying it could, take a year and you're hoping by the end of the year. So that's really encouraging to hear. I wonder if you could help us understand how J&J's approach differs from the messenger RNA approach we've heard from Moderna and the NIH, and also Pfizer today with BioNTech just announcing a partnership. Moderna is already starting human clinical trials that is going so incredibly fast. Just tell us about how your approach differs.
Yeah, the approach differs too. There is, of course, the time to clinical trials, or there is the time to get a real vaccine into the market, and there we can build on our existing platform. We have worked with the same platform, the same vaccine platform vector in HIV, in Zika, in RSV, and more recently in Ebola. And we have vaccinated in those fields now more than 50,000 people, shown that the vaccine is safe and that we have the proof of concept in animals now further into the clinic. And at the same time, in parallel, we have worked on upscaling, where we have the manufacturing sites ready to go, as well as the cell line, which can produce up to millions of vaccines. And so we can plug in the vaccine into an existing system, and that allows us to go very fast with very good information. And by now we know every hour, every day in this planning is already fixed until the product will come hopefully early next year, available the first batches for use in humans.
Kelly?
And, Meg, thanks. My question is, for people who have been exposed to coronavirus, can they get it again?
It's not clear. Normally, if you're exposed to a virus, you make the antibodies, you have the cellular immunity for the long term. It might be temporary protection, and you probably, like with influenza, need another a vaccine later on again, or you can get reinfected with a slightly different virus. So that all needs to be still studied. But you should assume that if you have now corona, that you should not be able to get it a second time, although I don't have the scientific data for that to tell you. But it's a biological principle.
Dr. Stoffels, it's Meg Tirrell again. I wanted to ask you also about your efforts to find a potential drug for the novel coronavirus. I think J&J said yesterday it does not look like HIV drugs will make sense to treat COVID-19. Tell us about that, and also the work you're doing to screen antiviral libraries of compounds, and how soon we could potentially see something there.
Yeah, there is some news on the antivirals for HIV, especially the protease inhibitors, and for our drug Persista. But we have not found the evidence yet that works, and we stay very close communication with the scientific community about that. There is no evidence in the clinic. There is no evidence preclinically, or even from the interface, the interaction of the drug with the virus or the receptors on the virus. So we don't think that will work, and unless that is proven, people should not use medicine for Persista for this indication. Now, on the other hand, screening of drugs, 15 years ago, when there was SARS, our company already made a screening system to get going on making a medicine. And we could reactivate that cell line now with the Wuhan virus and put it into a high-throughput phenotypic screening in collaboration with the university in Leuven, where they have a BSL-4 screening facility. That allows now to do in high throughput all the drugs in the market, and they could hopefully some of the drugs which currently are in the market, whether it are antivirals or other types of medicines, could have a direct antiviral effect. And following that, phase 3, phase 2, phase 1 drugs, those are currently already in the clinic, could be tested to make sure that if any of those has activity, they could go as fast as possible to patients. But it's using the logic that the drug has to be already in a certain clinical stage to be able to be used in a very short time. And that is in collaboration between many pharmaceutical companies. It's not Johnson & Johnson with support of the U.S. government, the European, and also the Gates Foundation is involved in that to make sure that we mobilize all pharmaceuticals as well as biotech companies to screen their drugs on activity.